Picture this. You're at the back of the bus, surrounded by all your friends. It's been a long day, so you're hot, you're tired. And that bean burrito you had for lunch is about to make a second guest appearance. You see, for the past several hours, your digestive system has been turning those little gems of fiber into methane, sulfides, and a host of other aromatic compounds. Sure, you think to yourself, I can be discreet. No one will ever know it was me. But wrong you are. Never has a group of friends turned on one of their own so quickly. So, come back to the lab with me, where we're going to investigate the science behind the age-old saying, "He who smelt it, dealt it." In biology, there's no such thing as a free meal. What I mean by this is that living things need energy in order to survive. Whether they're growing or reproducing or any number of other biological processes, living things need an influx of energy in order to continue to exist. As we've seen in previous videos, most of the energy used by living things on Earth actually comes from the sun. Autotrophs are able to use the process of photosynthesis to turn sun energy, or sunlight, into usable chemical energy, sugar, which all living things are capable of using. But the earliest living things on Earth, single-celled organisms, couldn't use photosynthesis to complete this process, and that's because photosynthesis hadn't evolved yet. These single-celled organisms needed to be incredibly choosy with how they spent their energy. They needed to find cheap and cost-effective means for which they could absorb nutrients and get rid of waste products. The process they eventually keyed in on, and still use to this day, is a process called diffusion. Diffusion is a process that's natural to our physical world. Through diffusion, particles, meaning atoms and molecules, move naturally from areas of high concentration to areas of low concentration. This is because all particles are in a constant state of motion. This is pretty obvious with things like gases and liquids, but even the particles in solids are sort of vibrating in place. They're moving around, just not very much, and that's part of what defines the difference between solids, liquids, and gases. This is why this motion, things like sugar can dissolve into tea, or we can use rock salt to melt ice. Even though they're solid, when the rock salt and ice come into contact with each other, the two sets of particles diffuse into each other and cause each other to melt. Diffusion is a process that does not require any external source of energy. It sort of happens for free, just because the particles are already moving. That means it's a process that living things are able to take advantage of with very little energy expenditure. They're able to use diffusion to power absorption of nutrients and elimination of waste products. Let's head over to the idea board to get a better idea of how diffusion works. As we mentioned earlier, diffusion is the motion of particles from a higher concentration to a lower concentration. Let's start with something we're familiar with: sugar and water. In this diagram, I've filled a glass with blue dots representing water molecules. As you can see, they take up space pretty evenly. They're not perfectly spread out because their motion is random, but they fill the glass overall, on average, evenly. Now I'm going to drop in a cube of sugar into this water. At first, all the sugar molecules, which are shown with red dots, are concentrated in the sugar cube. we can say that the concentration of sugar molecules is high here at the bottom of the beaker, but low elsewhere. The opposite can be said for water. The concentration of water is high everywhere in the beaker except in the sugar cube itself, where the concentration of water is 0. As time passes, the sugar molecules begin to spread out. This is caused by their natural random motion. We can't say for certain which molecules will move in which direction, but we can say that as a whole, or on average, they spread out over time. We can say the same thing for our water molecules. Our water molecules, which were in high concentration elsewhere in the beaker, are slowly dissolving into the sugar. And so, through their random motion, the water molecules are diffusing into the sugar. Eventually, the sugar molecules and the water molecules will spread out throughout the beaker until there's no longer an area of high or low concentration. At this point, diffusion doesn't have a direction anymore. However, it's important to realize that all these molecules are still moving around. They're just moving, on average, equally in all directions. They don't stop moving just because things have evened out. Their random motion keeps everything nice and even. So now you know how diffusion works, specifically the diffusion of water. In fact, this is such an important concept to biology that we give it a specific term, osmosis. We use diffusion to describe how all molecules eventually spread out over time, but when we're talking about the diffusion of water, it's so relevant to biology and how living systems work that we use the specific term osmosis to describe it. To help us visualize diffusion, we're going to dissolve some sugar into water. Intuitively, we know that sugar dissolves in water, but there are things we can do to speed up this process. By adding energy to the water, we heat it up. Heat is simply a measurement of the average motion of particles in a substance. Even though we can't see it, things that are warmer have molecules and atoms moving around much faster than things that are colder. Here I have two different sources of water. The one on the right is at a temperature of about 5 degrees Celsius, so slightly above freezing. And the one on the left is at about 60 degrees Celsius, too hot to put your hands into. I'm going to add about 10 grams of sugar to each at the same time. We'll put this on time lapse, and we'll see which one dissolves faster. The sugar has been in the water now for about 15 minutes. I'm going to give both beakers a quick stir, and we should be able to see that the sugar in the warm water has fully dissolved, whereas the sugar in the cold water is still mostly solid, although some of it has dissolved. If someone were to pass gas at the back of the bus, the immediate highest concentration is going to be right here at the back. In fact, the person who can smell it first is very likely the person who released it in the first place. But we know that those molecules aren't going to stand still. In fact, molecules in air move on average about 1,000 miles per hour. We don't realize it because those molecules have such a low mass. We just call that air pressure. But that's just the beginning for these molecules of gas. Randomly moving in different directions, they're going to bounce off of every surface, bouncing off of other molecules of air, slowly but inescapably making their way towards the front. You may think sitting in the front of the bus that you're safe, but no one can escape a trapped fart. So now we know that living organisms rely on diffusion to sort of transport their needs. But let's think of it in terms of concentration. Because of our metabolism, the chemical processes that keep us alive, nutrients are constantly being converted into waste products. This means that outside of our cells is a higher concentration of nutrients, and inside our cells is going to be a higher concentration of waste products. Our cells need to reverse that in order to stay alive. Diffusion lets those nutrients filter in and those waste products filter out of the cell. But there's a downside to this free process. Diffusion is slow. It's the reason why large organisms are made of many smaller cells instead of one single large cell. It's related to a rule called the square-cube rule, which regulates the relationship between surface area and volume. Let's head over to the idea board and we'll take a closer look at what we mean. Imagine you had a cell that was shaped like a cube, 1 centimeter to a side. Using some basic geometry, we can calculate both the surface area and the volume of that cell. The surface area comes to 6 centimeters squared, and the volume comes to 1 centimeter cubed, giving us a ratio of 6 to 1. This is a relatively high ratio, meaning that diffusion works relatively well for a low volume. However, if we increase the size of our cell by 1 centimeter, we can see that ratio goes down. A 2 centimeter side becomes a 24 centimeter squared surface area and an 8 centimeter cubed volume. If we increase again, both the area and the volume also increase, but the ratio between those two values goes down. That's because area and volume don't increase at the same rate. Area is a squared value, whereas volume is a cubed value. If we increase again, we see all of our values increase except for ratio, which continues to go down. And this is why cells can't get too big. Eventually, their ratio between area and volume gets so poor that they're not able to use diffusion effectively. Eventually, cells will either starve or they suffocate in their own waste products, which they are unable to remove. This next demonstration uses an agar gelatin cube, which has been treated with a chemical called phenolphthalein. Phenolphthalein is a pH indicator, meaning it changes color when exposed to acids and bases. In the presence of a base, it turns a brilliant pink. Bases such as sodium hydroxide can make that change. When exposed to an acid, such as hydrochloric acid, the indicator will change back to clear. We can use this effect to track acid as it diffuses through the agar gelatin cube. We're going to use 3 different measurements of agar gelatin cubes in this demonstration. We're going to use cubes with 1, 2, and 3 centimeter sides. And as you can see, that simple change in side measurement leads to a large change in overall volume. I'm submerging the agar gelatin cubes in hydrochloric acid. As the acid diffuses through the gelatin, it will change the phenolphthalein indicator from pink to clear. This should help us watch diffusion in action. Note that diffusion is not terribly fast, so we've had to speed up this recording by a factor of nearly 80 to fit it within this video.
Here's an agar cube that I stopped about halfway through the process. I can cut it in half and you can see more clearly that diffusion has happened from all directions simultaneously, but the center has not fully diffused yet because the overall size is too big. Diffusion is too slow once cells reach a certain size.
Large organisms get around this process by being composed of many smaller cells. Your body is made of millions of cells, perhaps only a few nanometers in length, billionths of a meter. But you've got a far greater surface area than you could possibly imagine. Evolution has guided our bodies to maximize surface area. For example, your digestive system has the surface area of about half a tennis court. Your lungs are about a full tennis court in area. If we think about your circulatory system, your blood vessels, which are responsible for providing nutrients and eliminating waste from your entire body, your circulatory system has a surface area just shy of 140 football fields. Our entire bodies are built to maximize diffusion. The other way that we can maximize diffusion is by controlling the concentration gradient. What I mean by a gradient is 2 concentrations that are next to each other. When a high concentration is next to a low concentration, there's a gradient between the two and diffusion will occur. If we can maximize the gradient, make it steeper, diffusion will happen for longer and more rapidly. But the reverse is also true, and we need to be careful of that. If that gradient flips, nutrients and waste will move in the opposite direction. The process of diffusion doesn't care what's inside or outside of a cell. The only thing that affects diffusion is the direction of that concentration gradient. So living organisms need to be very careful about what chemicals they expose their cells to.
Imagine this is a cell submerged in water. We can imagine that this is 100% pure water, which is the highest concentration possible for any substance. Inside the cell there's also water, but definitely not 100%. Cells contain many things beside water, so even if we don't know exactly how much there is, we know it's less than 100% water. Which direction should osmosis happen in? If you said into the cell, you're right. Remember, osmosis and diffusion always happens in a direction from high concentration to low concentration. It doesn't matter if this is 99% water, which is a high number. The higher concentration is outside and the lower concentration is inside. Water should move into the cell. When we're talking about the tonicity of a solution, meaning if it's hypo, hyper, or isotonic, what we're talking about is the relation between two things. In this case, we're talking about the relationship between a cell and the solution that it's been put into. This is an example of a hypotonic solution. The water, which we're imagining is 100% pure, has a higher concentration than whatever is inside the cell or the thing we're studying. Because the higher concentration is outside, water will diffuse in through osmosis. Hypotonic solutions diffuse into our subject. The opposite is a hypertonic solution. In a hypertonic solution, our outside solution is not as high as what is inside the cell or the subject. In fact, for this example, let's imagine that the cell is 95% water. Our solution is a saltwater solution with only 90% water. The other 10% is salt. Where is the highest water concentration? The highest concentration in this case is inside the cell, and so water will diffuse out of the cell through osmosis. This is a hypertonic solution, where the external environment has a lower concentration than the internal environment of whatever we're studying. But let's imagine that we use a less extreme solution. We create a saltwater solution that's only 5% salt, meaning the remainder of it is 95% water. Our cell is also made of 95% water. Where is the higher concentration? The answer is there isn't a higher concentration, and so water will diffuse out and into the cell in equal parts. This causes the cell neither to lose mass nor to gain mass. We call this an isotonic solution, meaning that both the solution and the interior of the thing that we're studying have a similar or identical concentration.
Today's experiment takes a closer look at everyone's favorite root vegetable, the potato. Deep fries fantastically, crunchy, starchy, great with every meal. But I digress. The potato is an excellent organism to study because even though this came off of a grocery store shelf, it's still a living thing and its cells are very much alive. We can cut up the potato into different samples and we can place those samples into different solutions. By creating different solutions of water and salt, we can experiment with different concentrations in relation to the concentration inside of the potato cells. We can create concentrations that mimic hyper, hypo, and isotonic conditions for the potato cells. In theory, if we put our potato cells into something that is hypertonic, that means that there's a low concentration of water outside of the potato cells, and the water will diffuse out of the cells, and our potato samples will lose mass. If we put our samples into a hypotonic solution, the opposite is true. The higher concentration of water is outside the potato cells, and water will diffuse into the cells, causing them to gain mass. If we've got a solution that is isotonic, it means the concentration of water inside the cells and in the solution around the cells is the same. The water will move back and forth evenly between the potato and the solution, but the potato will not lose or gain any mass. Let's take a closer look at how our experiment is set up, and then we'll run it.
Next, we're going to make a number of different solutions that we'll submerge our potato samples in. We'll be using salt to make different percentages of salt water solutions. And using water, this is actually very easy because water is the standard of all our measurements. 1 gram of water is equal to 1 milliliter of water. So if I want to make a 1% salt solution, that means 1% of the solution is salt and the remaining 99% is water. I create this by measuring out my 1 gram of salt and adding it to water until the total volume is equal to 100 milliliters. That gives me a 1% salt solution. If I want to make a 4% salt solution, I need to measure out 4 grams of salt and add it to water until I reach a total volume of 100 milliliters. I'll be preparing a total of 11 different solutions. The first one will be my control sample using pure distilled water, which I won't add any salt to. For the remaining samples, I'll be creating salt water solutions at concentrations ranging from 0.5% through 5% salt water. I'll create these at intervals of 0.5%, so my solutions will be 0.5%, 1%, 1.5%, 2%, and so on. Next, we need to prepare our test samples. Again, we're going to be using uncooked potatoes as our simulation live cells. I begin preparing them by cutting each potato in half. One of the important things that we need to do is make sure that each of our samples has the exact same measurements, and therefore has the exact same surface area. The potato coring tool that I'm using produces a cylinder cut out of potato with a diameter of 1 centimeter every time. Once I cut out my potato core sample, I also need to cut it to a standardized length. This will ensure, again, that all my potato samples have an identical surface area. We're going to be measuring our potatoes to a length of 5 centimeters. Now I'm going to add my solution to each sample of potato. We're going to let our potato samples soak for the next 24 to 48 hours. If we leave them sitting out that long, some of the water in solution will evaporate, but the salt will not. That will change the concentration of the different solutions. To prevent this, we're going to cover each beaker with a layer of plastic wrap. Alright, now it's been 24 hours, so we're going to take all of our potato samples out of solution. We're going to mask them and get to our final results. I'm going to take each sample out of its solution and let it drip dry for a moment to get rid of any excess moisture clinging to the surface. I'll then put them into a numbered weigh boat to keep track of them, and then I'll individually mass each sample. So here are our samples out of solution. In just a moment, I'm going to mass them using the electronic balance and a clean weigh boat. Remember, we always want to use a weigh boat when massing something on the electronic balance to keep the equipment clean and uncontaminated. And we want to zero the balance so we're not taking the mass of the weigh boat into account. Here's our first sample. Here's our second sample. And I'll continue the process. Now that we've got all of our data, we can do our calculations and figure out which solutions were hypertonic, hypotonic, and potentially which solution was isotonic. I've cleaned up our data table to make things a little easier to read. Now that we have our initial mass data and our final mass data, we can calculate the difference between these two values. Remember, we're going to want to subtract the initial mass from the final mass. We do this because this will give us the appropriate sign in front of the numbers. For samples that have gained mass, we'll end up with a positive sign. And for samples that have lost mass, we'll end up with a negative sign. Now we have the difference for each sample between its final and initial mass. We also have a sign in front of that number telling us if it either lost or gained mass. However, this value is a little misleading because every sample started out different. We need to find a way to standardize these values. So what we're going to do is we are going to calculate something called the percent change or percent yield. By taking the difference of each sample and dividing it by the initial mass, we're standardizing our measurements to show how each sample changed by percentage in relation to itself. If that sounds complex, it's really not. Here's the formula. Final mass minus the initial mass, which we've already done, divided by initial, and then multiplied by 100 to give us a percentage form. This will give us our percent yield or percent change. I'll fill in those numbers for you. I've cleaned up our final results so we can take a closer look at things and make some judgments based on our final data. Now that we have this percent change calculated, we can better compare these numbers. Looking at the first three, we can see that there was a positive change, and I'm going to add that positive sign in here. This means in all three cases, our sample gained mass. And we can see that, as we expected, our control sample, which was a 0% salt solution, or pure water, had the greatest positive change in mass. This meant that the solution was hypotonic in relation to our potato cells. There was a greater concentration of water outside the cells, and so water diffused into our samples, causing them to gain mass. So we can say that our first 3 samples are hypotonic. Everything from this point down lost mass to some degree. We can also see that most of our samples lost about 20% of their mass, although it's not uniform. This is largely due to the fact that we're dealing with living samples. Living things are very rarely standardized, so it's hard to get accurate measurements or even comparable measurements from living things. If we wanted better measurements, we would probably have to repeat this entire experiment several more times and average out our results. But what we can see is that they all lost mass. This tells us that water in the potato cells left the cells and went into the solution. This means the solution had less water than the potato cells, and so we can say that these solutions were hypertonic. And I'm just going to add an arrow in for that. But which sample is isotonic, or which one is closest to isotonic? Well, sample 3 had the least change in mass, either gain or loss. It only gained about 2.5%. So we can say that potato cells are isotonic with about a 1% salt solution. It's probably a little bit between samples 3 and 4, so isotonic is probably somewhere between these 2 solutions. If we wanted to get this more accurate, we could repeat the entire lab using much smaller gradations between our sample solutions. We could do 0, 0.1, 0.2, 0.3, and so on, with smaller and smaller solution types in order to get a more accurate reading. We also now know the effect that diffusion and osmosis can have on living cells. It allows cells to move materials around cheaply, nutrients in, and waste products out. But it does have some limits. As cells get larger, they eventually either suffocate or starve because diffusion is simply too slow. But not all cells are created equally. Your skin cells, for example, are about 40 nanometers across, and even though that's small, it's too wide for diffusion to work effectively. They get around this by being incredibly flat, like little pancakes. Each skin cell is only about 5 to 10 micrometers thick, and that is small enough for diffusion to function. But there are more extreme variations out there. An ostrich egg is considered by scientists to be the largest single egg by mass, coming in at about 1,400 grams, or about 2 dozen chicken eggs. It gets around the problem of diffusion by not needing it. You see, when an ostrich lays an egg, all the nutrients are already inside the cell, and as the growing chick develops, the waste products are stored in a separate portion of the cell, so the chick isn't poisoned by its own toxins. However, if you're looking for the longest cell, look no further than nerve cells. Nerve cells are incredibly long, but they're also incredibly narrow, and that's why diffusion still works with them. The longest known nerve cell is the dorsal root ganglion, which goes all the way from the foot to the brainstem at the base of the brain. In humans, well, tall humans, it can reach about 2 meters in length, but that's nothing compared to the blue whale, the largest animal ever to exist on planet Earth. In the blue whale, the dorsal root ganglion can reach lengths up to about 25 meters, or about 75 feet. That's so long that nerve impulses can take upwards of 6 seconds to travel from the source of impulse at the foot or tail all the way to the brainstem and back. This imposes a whole new set of restrictions on organisms, not on cell size, but how big the organism as a whole can get. How might organisms get around these limitations? The answer to this is responsible for much of the diversity in the world around us. But more on that another time. Until then, stay curious, friends.